And next up we have Kresten Lindorf-Larsen from the University of Copenhagen, and he'll be telling us about understanding the origins of loss of protein function using analyses of protein stability and conservation.
Thank you very much. Thanks everybody for organizing this fantastic meeting. So I'd like to tell you about some of the work we're doing in Copenhagen trying to scale computational methods to study all of these small dots which are proteins. But also I think one thing that I hope to convince you is that we can also maybe use computational methods to understand a little bit about what's going on. So not just focusing on prediction, but also understanding mechanisms. So I'm working with a bunch of people, and in particular I'd like to mention two of them, Rasmus Hartmann and Amelie Stein. So everything I'll talk about today is a collaborative work with the two of them, but we also have a center that involves Doug and a couple of other people. And we're working on several different things, but in particular I come from a background of protein folding. Rasmus comes from a background of protein homeostasis and degradation. So we're sort of trying to see some part of all these missense variants that we'll look at, and I'll talk more about it in details, simply work with a very simple mechanism that they destabilize the proteins, they get degraded, and therefore you lose function. So we'll try to figure out, can we predict these and can we understand them? And can we learn something about what's going on using both normal cell-based assays, but in particular MAVES. So we've done several things that I won't have time to talk about, but I'm happy to talk about it later. We've recently scaled up these kind of calculations to more or less, not the entire proteome, but a good fraction of it. I won't have time to talk about this. We've actually looked quite a bit at what governs protein degradation, so mapping quality control diagrams, which I won't talk about, but there's a preprint out. And then we're using VAMP-seq data to try to understand the distinction between cellular abundance and protein stability. So I'll talk a lot about stability, but actually what I'll really talk about is abundance, and they're related, but they're not the same. But we're trying to figure out what the differences are. I won't have time to talk about it. I also won't have time to talk about 4 posters work from our group. So these 4 people, you've seen some of them before, are presenting maps of 4 different genes. Glucokinase, that's a preprint out, aspartate Parkin, and Sophie is going to present some data from her work with Amelie Stein on dihydrofolate reductase, and I won't have time to talk about it, but again, go check their posters. I'll talk about some other things. And so something that is, maybe not a surprise to all of you, right, but human genetics provides information about, people, but also molecules. And as a biochemist, the molecules are important to me. And so when I see, pictures like this, it makes me slightly unhappy, right, these kind of weird helices that twist the wrong way. Right, that picture, that thing on the top left, that's not a genotype-phenotype map of, the handedness of your DNA and which side of the road you drive your car. It's instead, a molecule that isn't really what it's supposed to do, right? The molecules are important to biochemists, and so we'll try to sort of dig in and look at the molecular structure rather than, pretend that these are just, abstract phenomena. So that will be extremely simplistic. This will be, a little bit more fancy than this, but not quite. But all right, we'll really simply try to look at, proteins that have an equilibrium between the folded and non-folded state, and then we can ask questions like, can the mutations shift these equilibria, and can we somehow correlate it with things that we can observe in cells? And we can do measurements of these things, and we can also do predictions of these things. So I won't actually talk about the measurements, but Sophie, shown here, did a lot of quite nice measurements on MSH2. I'll show you some of our computational work, and it's pretty simple for us now, and many of you have probably done this, right, to do a saturation scan of a protein, but just calculating the effect of each individual amino acid substitution on the thermodynamic stability of the protein, and then we can try to correlate these things with measurements of in vivo abundance, and as I said, they're not the same, but they are related, but I won't actually have time to show Sophie's nice data. I'll just show this picture on the left, which is sort of, I think, a nice example of a set of variants where we can pretty well distinguish pathogenic variants from non-pathogenic variants simply by whether they destabilize the protein. And so if we look at sort of a ROC curve analysis, for example, you can see that this very simple method of predicting thermodynamic stability actually classifies variant pretty well. Not perfectly, but pretty well. This is a little bit old, so there weren't so many high quality variants at the time known in MSH2. Amanda did a very, very similar study on another protein, MLH1, that you also heard about yesterday from Jacob Kipling, and we found something very similar, mostly that there's a bunch of disease variants that are highly destabilized, and we showed that they get degraded by the proteasome, And so we can classify all those variants pretty well, but you will also see this, some pathogenic variants that are, found at normal cell levels, right? And this, will be the topic of the next few minutes. So for anyone who does anything with biology, right, it's clear that you can lose function beyond just losing the protein, right? Proteins are complicated things. They do many things, and one of them is that, most of the proteins need to be stable to be functional, but you can lose function in another way. So I'll show you a bunch of maps that look like this. Sometimes, unfortunately, we're swapping the axis a little bit, but on one axis, I'm showing you the delta G, which is the predicted change in thermodynamic stability. So on the right, proteins will be unstable, and the other axis, we're sort of analyzing multiple sequence alignments using a statistical model, very similar to what Mafalda, I think, we'll talk about later. So things at the top are essentially substitutions that are sort of scored to be bad by evolution. So at the top right, all those variants, and those were all disease-causing variants, in this case of MLH1, they are predicted to be thermodynamically bad for the protein, and evolution say, well, this is a really bad idea to put in a proline here. At the bottom left, these are essentially all benign variants. Evolution says, well, that's absolutely fine. You can replace this leucine with a valine, the protein will be happy, and the stability predictions say so also. And then there's the variants at the top left. So these are variants that are pathogenic, not because they are degraded, thermodynamically, they're just fine, but they're doing something else to the protein, right? They're somehow perturbing the function. So by analyzing jointly thermodynamics and evolution, we can sort of try to decouple these effects of, stability things versus non-stability things. So I'll try to elucidate that, in a few more slides. So clearly, if we just want to predict whether something is pathogenic or not, right, there's no real good reason just to focus on stability, right? And so whenever we try to look at, conservation scores, they're typically better predictors of pathogenicity and just stability because it will capture both the things that are unstable, but also the things that are stable but lose function in various ways. But by contrasting the two, we can actually try to disentangle mechanism a little bit more. So if you're interested in just, big quotation mark, obviously, predictions, evolutionary methods are fantastic. We use them a lot. If you want to understand mechanism, they really often don't tell you that much, right? They just tell you that this is important for the protein, but not really why. So just before I go into, I just want to show this slide, and it comes up with something that has come up several times here. This is now, we've taken our predictions on MSS2 from 2017 and sort of put them on top of the map that J.P. Kitzman showed you yesterday. So I'm showing you here a distribution of the scores from the deep mutational scanning experiment superposed with some average scores from our predictions of this delta E, which is the evolutionary score, and the delta G, which is the stability score. And what you will see is that obviously, not surprisingly, right, the pathogenic variants, the ones with the positive score in the MAVE, right, they have high delta Gs and high delta Es, and the ones on the left have low values. But you will also see that there is a gradual change within the big thing that we would normally consider to be sort of wild-type like, right? So the ones with a score that's closer to 0 on the right, have a different evolutionary score and a different stability score than the ones on the left. So there is a gradual shift between those two scores. So this, it's not that everything from -7 to -1 is the same, but they're really different. So I think these computation methods are really supporting this idea that we can really distinguish between things that are very bad, a little bit bad, and perfectly fine, right? And I think these kind of methods will sort of support each other. And of course, it's also nice to be able to go back and validate your predictions. So one of the tools that we are using a lot is MAVE data to try to benchmark and understand things. So we were lucky to have PTEN had 2 MAVES, one probing activity in a growth-based assays, and then Doug published a really nice paper on the VAMTech tool. So we tried to say, can we sort of use these kind of MAVES where we have two different pieces of information to try to disentangle these a little bit more. And then because we sort of come from a more prediction background, say, well, maybe we can correlate them, as I've been saying to you, with these kind of sequence-based methods that we say, well, maybe they capture something about function. And then the stability things, they really capture things, related to abundance. So Mikael analyzed this data and tried to see, which of these things would be good at classifying variants, for example. And again, I'm not giving you many details, only that if you take some set of pathogenic variants and a reasonable, things that are sort of not quite benign, but sort of nomad variants, it turns out, maybe not surprisingly, that the best thing you can have is an experimental measurement of activity, right? If you want to classify, that's the best thing you can have. And then you can make 2 choices. You can either say, well, I want to replace my experimental, probe of activity with either an experimental probe of abundance or a computational proxy for activity, and it turns out that those two are about equally well. Right, so we can either take the VAMSEC assay that does prediction pretty well, or we can take these evolutionary scores and that works about as well. And then the worst we can have if we just want to do prediction is to predict the stability. But what you will also see in this ROC curve and some of the other ones also is actually we can actually classify many variants extremely well from abundance. Right? So the ROC curves go up quickly, and that's because there's a bunch of variants that are very easy to predict because they're just gone from the cell, and then essentially we lose information because the rest of them, they lose function by other mechanisms. And I'll get back to what they are. So we sort of built upon this work, or Matteo built on this work, trying to understand more generally what's going on, what are these different variants doing, and so again we looked at these kind of two-dimensional sort of multi-readout MAVES. We have multiple proteins now, but I'm showing here data for NOT-T15 and PTEN. So again, now I'm swapping the axis a little bit, but the variants at the top right, these are the ones that are active and found at wild-type levels. The ones at the bottom left, those are variants that are inactive and found at low levels. So we expect that they probably lose function because they're just gone from the cell. And then the variants at the bottom right, we call them blue residues. They are the ones that are present in wild-type amounts, but are inactive. And we've looked at several different systems, and it turns out that there's typically about 50-50, maybe a little bit more in the bottom left compared to the bottom right, suggesting that maybe 60 to 70% of the variants, they lose activity because they are gone from the cell, and then about 30-40% of the variants, they lose function but are found at wild-type levels. So this we get from the MAVE data. So before we go into the predictions, we can sort of go back and we can try to understand what's going on. And it turns out, and this turns out to be useful, and actually these variants at this bottom right corner, so these are variants that are found in the cell, mutations at those sites, perfectly normal wild-type levels, but they're inactive. If you map them onto the structure, they all cluster in functional regions. Right, so this comes back to something we heard yesterday, right, that if you combine measurements of activity and abundance, you can really learn something about active sites. Right? So all of these blue residues are things that we color-coded simply from the MAVE data, and we can see, well, they're really clustering in this case, the active sites of enzymes, but as I'll show you in a second, also other things that you can call allosteric sites, but certainly sites that are important for function in one way or another, what the cell is probing. But before we do that, let's also just go back to our predictions. And I mentioned again, this thing that we have two different prediction methods. The plot on the left for NOT-T15 is a little bit cleaner. So we correlate stability predictions or conservation scores with these two different MAVES. And maybe not surprisingly from what I've told you so far, right, the stability predictions correlate clearly better with a VAMSEC score, whereas the evolutionary scores, the evolutionary predictions, they correlate better with an activity measure. So that suggests that we can use predictions, stability and conservation as proxies for MAVES that probe activity and abundance. And that allows us, of course, to do predictions. So Magnus started doing this work, so we started essentially to taking a bunch of data where we had proteins with known structure, we have MAVE data, we tried to sort of do these kind of calculations just to see whether we could scale up things. It worked pretty well. These are the same kind of maps that I've shown you before, right? On the x-axis, stability. On the y-axis, top things are things that evolution would think is a bad idea. And it's color-coded, with blue variants being things that are active in some kind of MAVE. And you can see all the things at the bottom left, perfectly fine protein. Things at the top half are variants that lose function. The ones at the top right, they lose function due to loss of stability, and the ones at the top left, lose function due to other reasons. And again, sort of 50-50, maybe a little bit more on the right-hand side, depending on how we put the color, right? So we're seeing this very general picture across many different proteins that, maybe 50, 60, 70% of the variants that cause loss of function, they do so simply because the proteins are unstable. So what Matteo then did, and this is unpublished work, but soon hopefully not unpublished anymore, is that he took these MAVE data and tried to build a little simple classifier. Can we sort of plug in stability calculations and evolutionary scores, train it on these kind of two-dimensional MAVES, and see can we classify variants into those that lose function due to abundance and those that lose function due to other things, so that we can predict what's actually going on and maybe useful in terms of interpreting variant and variant mechanisms. So that turns out to work pretty well. I'll just show you sort of a little bit of a random example, but with some nice complications of this protein is doing interesting things. So this is a protein that forms a complex. We just do stability calculations on the monomer. So that means that mutations at protein-protein interaction sites, they don't affect the stability of the monomer, but they're clearly conserved. Right? So we can pick up those. Those are the blue residues at the bottom right. Residues in the active site, light up beautifully, right? We can see these are active sites, but we also see, again, coming back to things we heard yesterday, right? We see sort of second shell kind of thing, right? Things that are really nicely conserved, but not due to effects on protein stability. So we can really, in sort of a relatively unbiased way, pick out regions that are important for function in a way that is not polluted by sort of stability effects. So we've done this on quite a few proteins. I'll just show you one example here that's relevant for human disease. So this is HPRT1 that many of you will probably know, right? It's involved mismatch repair. Missense variants are involved in Lesch-Nyhan syndrome, a disease you maybe don't want to Google if you don't like bad images. So Matteo went through this kind of exercise on this protein, and Rasmus and his lab, they picked out we picked out a bunch of disease variants, asking can we use these calculations not just to classify variants into disease or not, but also why and how they lose function. Right, so we have an assay that proves both function. This is a yeast-based growth assay. And abundance, this is all in low throughput, just with Western blots. And it turns out that we actually can classify these things pretty well. Right, so we can predict which variants cause loss of function, but we can also predict which variants cause loss of function simply because they're gone from the cell versus which ones are just inactive. And so again, this can be useful if you're interested in the biology, but of course it could also be useful if you're interested in sort of rescuing these things. If you want to bind something to restabilize, that's only going to work if the reason why you lose function is due to loss of abundance, but these blue variants cannot be rescued. How am I doing for time?
4 minutes left.
4 minutes. Okay, then I'll just very, very quickly mention one other thing that we can do with these MAVES. And this is, sort of the flip side of things, so maybe not always so relevant for human disease unless you try to do engineering of, for example, antibodies and stuff. And it's very related to things we heard about yesterday. So this is work from Christopher Johansen together with Jakob Winter. And so again, the idea being that, if stability is such an important readout, maybe we can learn something about stability, maybe even make proteins more stable. And the problem is, in many of these assays, we're already saturated at the beginning. If you start with a stable protein, it's very difficult to, in your readout, find things that are more stable. So we're building on earlier ideas, from some of the people here, on sort of finding sort of internal suppressors. But instead of, looking at single double mutants, we sort of just say, we have a big library of multi-mutants. We do a global analysis, and we sort of find things that in a background of some other variants can restabilize. So in the middle plot I'm showing you here, this is just titrating GFP with mutations. You see you get less and less green protein because it falls apart. If you then sort of disentangle that and, for example, look at the effects of titrating in a specific background, for example, this red curve up the top is a valine to alanine mutation, you can see that you need to put in more mutations to destabilize the protein if you have that valine, meaning suggesting that this valine is probably good for the protein. So you can do this analysis, you can essentially estimate single variant effects, assuming an additive model, and we can discuss that afterwards. And then you can find relatively subtle effects, but because you've oversampled your mutation space and because you're sort of moving the dynamical window with this kind of titration, you can sort of find things that actually make the proteins better. And so these are things that we wouldn't really have been able to predict. They're sort of located on the surface, but they make the proteins, in this case, more green. So we've actually taken some of these top hits, put them back into GFP. So the figure on the left, the one says as wild type is our starting point. If we take the top 3, 6, or 9 variants, actually we get much greener cells. And then at some point we start to run into problems, of whether that's due to epistatic effects or not, we don't really know. And what we also see is that we couldn't have predicted these with standard prediction methods from, say, Rosetta or evolutionary scores. So we can fish out these beneficial variants that we couldn't see because we were saturated in the dynamical window. But by essentially doing a global analysis of multi-mutants, you can pick them out. So this is one example. There's just a one-slide summary of a whole study done by Rasmus Noel in bacteria with a sort of a tripartite folding sensor, taking a protein, a designed protein, extremely stable. There's no way that you can get a readout in a normal assay. You mutate the hell out of it, you do this analysis, and then we took this protein that had a melting temperature of about 100 degrees and increased the stability to 150 degrees or by 50% in the delta G, right? So again, this kind of idea that we can do these kind of global models and get information about stability that we can then put back in and make proteins more stable. So with that, I'd like to end, summarize, hopefully have convinced you that protein stability is an important part of human disease. We can understand and we can predict it. We don't really know the relationship between stability and abundance, but we're trying to figure that out. And many other people are. We need more data. And then with these kind of multidimensional readouts, we can sort of start to disentangle effects of activity versus abundance, and that allows us to say interesting things about where the active sites are, what are allosteric sites, but also maybe think about how we can sort of manipulate proteins in different ways. So with that, I'd like to thank you for your attention and thank the people that did the work, in particular Emily and Rasmus, my partners in crime, and then a bunch of people that I mentioned along the way. Thank you.
Hi there. Yeah, thanks for this great talk. Have you applied any of these methods to proteins with disordered regions and ray stability, or maybe not stability, but abundance? Can you make any conclusions?
We have not done that, no. What we have done, and this is the work that I didn't talk about, we've done, or actually our collaborators in Israel has done a piece of work where we essentially put peptides onto a reporter gene to find out what's degraded, and then we know what the signals are. And so in those cases, we've mutated them to increase abundance of these, essentially remove the degrons. And it's pretty simple. If you're hydrophobic, you're degraded. If you put in negatively charged, you're not degraded. So this would be an important factor for abundance of IDPs. And in fact, we've shown that if you look at just abundance measurements of proteins with disordered regions, this correlates with essentially the hydrophobicity and the lack or presence of negatively charged residues.
Thank you.
Just want to make sure I heard that right. So you said negatively charged amino acids protect?
Yes.
Okay, so what are the implications for transcriptional activators? So do you think it's like they're...
I mean, I don't know, but it's obvious to speculate that one of the reasons why they have negatively charged is to prevent them from degradation. But we've not studied that in systematic details. And there's some data, we've looked at it a little bit, but that's, I think, likely that's one of the things that what those acidic things are doing is
because they tend to have both hydrophobic and negatively charged acidic amino acids. So is there a correlation? So more hydrophobic you have, you need more protection?
We've not looked at that, but I think the negative charges are also doing something functional. And maybe also protecting, not protecting against binding to DNA, but I also think that they're preventing degradation. But we've not looked at that. But there's a lot of nice data. People not controlling so much for abundance, more like looking at transcriptional activation. But I think this is something to look into.
Beautiful.
Absolutely.
Thank you.
So all of this is spectacular, but has this very two-state view of protein folding and protein structure. And so with your unique perspective on ensemble determinations of proteins, what's the roadmap for sort of a more complex energy landscape on the folded side of things being integrated into this for you?
That's a very good point. And so first of all, we find that most proteins, if you peel off, let's say, a couple of kilocals per mole, they will be degraded, which is much less than the typical thermodynamic stability, which is already telling us something. So I don't think that these are global unfolding events, and that means that our stability calculations are probably correlated with something that isn't global unfolding. So our pathways are multiple. One of them, we're trying to convince people to, we've done a little bit, but try to probe this with multiple mutations. So the hypothesis would be that if you put in two mildly destabilizing in a thing that locally unfolds, they would be additive, but if you put them in two different regions that only do the independent local unfolding, you would not see degradation. On the computational side, we want to do things that we can scale, which means that molecular simulations are not the way forward. We've done some molecular simulations of unfolding of dihydrofolate reductase where we have hydrogen deuterium exchange to correlate with, but I don't see that as a long-term pathways. We are now, what we're trying to do, and this is, again, things that I haven't talked about, trying to find all the outliers in the VAMP-seq data compared to the stability calculations and see where, what is being degraded, even though they're not very destabilizing, or vice versa. Can you destabilize the region, but it's not degraded, possibly because you don't have degrons? So we have some ideas on how to do simple models of unfolding, overlay it with the degrons, because clearly these things are degraded, and then try to put that into a coherent model. But we don't really have a solution yet. But this is the roadmap at least.
Okay, let's thank Kresten once more.
Thanks.